Hello class, today we're talking about the reproductive system, chapter 27 from our textbook. So broadly, the job of the reproductive system is to produce and deliver the gametes. And so for a male, that will be a spermatozoa. For a female, that will be the ovum or egg. So we have two types of organs in the reproductive system: the primary that actually produces the gametes, and the accessory that is actually applying some sort of supportive role. So either acting as a gland for accessory fluids, or perhaps acting as some type of muscular tube to actually deliver the product. So this is hormonally controlled, and going back to our endocrine knowledge, these are regulated by tropic hormones. Tropic hormones being endocrine hormones that actually control and regulate other hormones. In this case, it is GnRH, gonadotropic releasing hormone, synthesized and secreted by the hypothalamus. This will tell the anterior pituitary that it needs to release this follicle stimulating hormone and the luteinizing hormone. These two hormones act on the gonads to regulate the production of our sex hormones. In this case, our major androgen being testosterone, which is largely a male sex hormone, and then our estrogens, the estradiol and progesterone. And so both sexes actually have both testosterone and estrogen, but the levels will vary. So this is controlled by a very typical endocrine axis. You have the signal being sensed at the neuroendocrine integration site that, for whatever reason, the sex hormones in the blood are low, and so what will happen is the hypothalamus will secrete the GnRH. That will induce the anterior lobe of the pituitary to upregulate production of the LH and FSH, and then those two hormones will work on the respective gonads of the sex. So if it's a male, that will be testes. If it's a female, it will be ovaries. And then as these sex hormones are made and the concentration increases via negative feedback loop, this will actually be slowed down. So one of the issues we run into with gametes is that they are derived from actually diploid cells, while gametes themselves are haploid. And that makes sense when you start to count the chromosomes, right? You get 23 from each parent. Imagine if you had gotten 46 from each parent. Well, each generation would have 92 chromosomes, which of course is not how that happens. And so we use something called meiosis actually to take copies after the S phase. After the cells go through S phase, they're copied essentially, and we're taking those copies and then splitting them in half and reducing that number of chromosomes. We call that reduction division. And then after reduction division, we need to divide again into the individual daughter cells. In this case, we're taking, looks like we're taking the diploid cells and splitting them into 4 haploid cells. And so in meiosis 1, there is a stage where crossing over can occur. And so at this point, these different chromosomes will exchange DNA sequences, and if the crossing over occurs, what will happen is there will be a sort of exchange of DNA that leads to genetic variation. If crossing over occurs, then all 4 of the daughter cells after meiosis 2 will have a unique DNA. If crossing over does not occur, and it doesn't always, then instead you'll have 2 pairs each of 2 different types of DNA. And so this is what that reduction division is illustrating, that you actually do have to go in and reduce those diploid cells to haploid cells. Otherwise, you'll end up with some sort of nonsensical chromosome count, like 8N, rather than the 2N that you're supposed to have. It's just an incompatible amount of DNA. Now, humans, being sort of these more complex animals, higher animals, we handle these events very poorly. But some organisms, like plants, actually do have these chromosomal doubling events and can withstand it. They can evolve with that essentially into some new organism. So meiosis in general is taking a diploid body cell and making a haploid gamete from it, and then also introducing the genetic variation from crossing over. So jumping right into reproductive anatomy, we'll start with the male side. So we have an external and internal side. In a male, the external reproductive organs include the gonads, accessory glands, and the ducts. And so the gonads are where the external reproductive organs are the scrotum and the penis, while the internal are the gonads, accessory glands, and ducts. Gonads, of course, are the site of sperm production and the site of the actual synthesis of those sex hormones, testosterone for androgens. The accessory glands are producing these supporting elements, so things that will nurture the growing sperm, things that will help the sperm move out of these ducts, and while the ducts themselves are actually carrying the sperm and the glandular secretions to where they need to go. So starting in the internal, we have the testes. Sperm is going to be produced in the seminiferous tubules located within the testes. And this process is very highly regulated in terms of temperature. Sperm actually does not develop well at body temperature. And so the purpose of the scrotum is actually to contract and expand in order to keep the testes at an optimal temperature. And it's actually about 3 degrees Celsius below that 37-degree core body temperature. And we call a testes located within the scrotum a testicle. And of course, in a healthy male, there will be 2. So let's talk a little bit about the ducts that are actually carrying the sperm and the accessory fluid. So there is a coiled duct coming from the seminiferous tubule. You will pass into this epididymis. And so this is where the sperm will complete maturation and eventually become motile. We'll see in a second that there is a lumen where you can look and see fully formed sperm, but they don't yet have the motility needed to get where they need to go. And so looking at the ejaculation pathway, moving through here, first you'll come off the vas deferens, move through the ejaculatory duct, and then eventually the shared exit of the urinary bladder, the urethra. And so we'll see in a second that one of the jobs of the accessory fluid is actually to neutralize the pH of the urethra because it also uses that as a pathway for urine. So what is semen then? Well, semen is a collection of things. It's the sperm, it's the secretions from these accessory glands, and basically it serves as a nutritional media for the semen or for the sperm, and also acts to lubricate the glands and the ducts, and also serves to neutralize the pH in this area. So for the accessory glands, you have 2 seminal vesicles. About 60% of the total volume of semen is coming from these, and this is the transport media. Then you have the prostate gland. Prostate gland is secreting directly into the urethra, and this is a protective medium, and it's also somewhat nourishing for the sperm. And then you have 2 copies of the bulbourethral gland, and so this is sort of the mucus-based composition of the semen, and it's essentially coming before the sperm in order to make sure that that urethra is the correct pH, because the sperm themselves, of course, are using lots of proteins, and you don't want those proteins to be damaged by the pH of the urine, which has passed through the urethra before. So looking at some of this external anatomy now, we have the penis. The penis is actually just a series of spongy cylinders, essentially, that can fill with blood. And during arousal, these erectile tissues are going to just fill up with blood, and then there will be some kind of signal closing off the veins just to help to maintain that erection. Then looking out towards the glands, the head of the penis, we'll have a thinner skin covering than the shaft, and then naturally we have a foreskin, but this tends to be removed in some countries when a male is circumcised right after birth. Okay, going back to the testes. Remember, when we talk about outer coverings, we tend to use the word tunic. So, for instance, vasculature has tunics, the outer surface of the eye has tunics, so does your testes. Your testes has the tunica vaginalis, which is that outer layer, sort of derived or continuous with the peritoneum, and then the tunica albuginea, which is the inner layer and forming a more structural fibrous capsule. So the testes themselves will be subdivided into these little lobules, about 250 lobules each, And then each of these lobules will contain 1 to 4 of the seminiferous tubules. Remember, this is the site of sperm production. So tracing this path all through the seminiferous tubules to the epididymis, you have the straight tubule, the reedy testes, the efferent ducteals, and then eventually you end up in the epididymis. And so the epididymis is itself sort of this coiled duct, and it will have a head, a body, and a tail. And this is the site of sperm storage until ejaculation. So in general, just sort of in review, the testes is the site of male gamete production, so it's the gonad essentially. The seminiferous tubules, these highly coiled, lots of surface area, packed tightly into these lobules. Then you have a scrotum, which is holding and maintaining that optimal temperature for sperm maturation. And you can see if it's too cold, it will tract. If it's too warm, it'll relax and sort of move away from the body. This is being accomplished by the cremaster muscle, which is actually a smooth muscle. Okay, so we get to the seminiferous tubules, of course, the site of spermatogenesis. We're going to look at some different spermatocytes here. Starting on the outside edge, we have the spermatogonium, and this will have the both copies, the red and blue DNA. And then moving into further towards the lumen, we're going to have a primary spermatocyte. Also has 2 copies of the DNA, but after this, to get to the secondary, we're actually undergoing meiosis 1. Moving into the early spermatid, where we've undergone meiosis 2. So we've completed our reductive division and our equational division. So now we are left with immature haploid sperm. And assuming crossing over has happened, you should have 4 different genetically variant cells. And then as it continues to mature, the sperm will actually start to change shape. It will turn into that sort of characteristic torpedo shape that we know. It will lose sort of that outer covering. It will get a chromosome, but sort of getting ahead of ourselves. But as it, the point of this slide is that you're starting on the edge of the seminiferous tubule and then migrating towards the lumen as you continue to mature. And eventually in the lumen, you'll be left with these mature sperm that have yet to develop motility. And so some other cell types that you see in this area include the Sertoli cells. These are essentially nurse cells that are feeding and nourishing and caring for those developing sperm as they make this journey through the cross-section of the seminiferous tubule. All right, so as we have moved from the seminiferous tubule cross-section to inside the lumen, now we're going to see the cell actually start to take that characteristic shape, right? So it's got a nucleus. This is the site of where all of the DNA is actually located. It's got a cytoplasm just like a cell would. The chromosome will begin to form. This is sort of a protein cap. Then the cytoplasm will begin to fold as the flagellum emerges. Finally, that flagella is going to emerge completely. The cytoplasm and the mitochondria are all going to sort of move towards that flagellar end, that pole. The mitochondria, of course, there because when the sperm gains motility, it's going to need a bunch of energy to run that little tail. And so eukaryotic flagella whip back and forth. This is kind of in contrast to bacterial flagella, which tend to, I think, spin in one direction, but lots of mitochondria in a sperm cell, but no other organelles. So we have a very high ATP need in this cell, but not much else is going on. And so our final sperm has a tail, a midpiece, and a head. And then very much a quantity over quality reproductive strategy for males. But when we look at females, it's not quite the same. Okay, so in the lumen, the sperm are going to be complete, except they do not have motility yet. And so motility is going to happen as the sperm are actually pushed through these various ducts, the epididymis and the ductus deferens. And this kind of explains why we need a little more surface area here. If you look at the ductus deferens, it actually hangs over these structures here, over the ureters. And this is just giving the sperm enough time to actually get time for the sperm to gain motility. And so that is why they're so highly coiled here, because as we push the sperm through, they just need time to actually develop. So looking at the urethra is running the length of the penis. It's almost entirely erectile tissue. Of course, this is also where we expel urine from, so that has to be accounted for with our accessory glands providing some support. And basically what happens during erection, a parasympathetic signal will cause the release of nitric oxide. This will dilate the arterioles and the erectile tissue filled with blood. That is a short-term signal, and then you'll have these sort of sympathetic signals that help to keep the veins sort of contracted so that this erection can maintain. So the expanded tissue will press on the veins, which will help to close those veins off, thus the blood stays in the erectile tissue. So our initial trigger is parasympathetic. Then you have a sympathetic nerve signal firing to maintain arousal. These excess signals cause the glands and ducts of the semen to push the semen into the urethra, and then rhythmic smooth muscle contractions will actually lead to the ejaculation itself. And so semen is 70% seminal fluid and 25% prostatic fluid. The sperm itself actually makes up a very small amount, perhaps only 2 to 5%, but when you think about it, these are very small cells, and there could be up to 500 million sperm per ejaculation. And so the whole point of ejaculation is to expel the sperm outside of the body in the form of this semen. Okay, moving on to female reproductive anatomy. The external anatomy is going to appear very similar to the male anatomy. That's because the male structures are actually derived from the female structures. There is a point in development upon which all people are actually female, but the internal organs, again, you're going to have a gonad, in this case an ovary, and a system of ducts that will carry the gametes. The difference is, of course, you actually need to host the embryo in this case so that it will develop. Okay, so we get to the oviducts in the uterus, and essentially we need to start off with the process of ovulation, essentially releasing a mature egg and moving it from the ovary where it developed to the oviduct, also called the fallopian tubes. Cilia in this area are going to convey the egg to the uterus, sometimes called a womb, and the womb has a sort of vascularized inner lining called an endometrium, and so this vascular lining is actually going to help feed that egg cell. So this will connect via the cervix to the opening of the vagina. So moving to the vagina itself is a muscular elastic chamber that is simply acting as a receptacle for sperm, but also as the birth canal when the fetus is actually being born. That external opening would be the vulva, and it consists of the sort of external organs that we had mentioned before, the 2 labia. There's a hymen that will remain until penetration, and then the clitoris itself. So, moving on to female gonads. The gonads of the female are referred to as ovaries. Ovaries are actually going to lie a little bit higher than you would probably guess, up in the abdominal cavity. And when you look at their form, they have a cortex sort of on the outer side, and on the inner side is the actual medulla, which will have the vascularization, bringing in nutrients, as well as the nerves sort of translating these nervous signals. Each ovary is going to be sort of inundated with these follicles, and not all follicles are actually going to mature over the lifetime of the female, but these are the tiny cyclic structures in the cortex where the oocyte is actually located. And so the oocyte will actually be encased in a layer of different cells, like granulosa cells we'll see. But once a month during their cycle, a ripened follicle will be ejected in an event called, sorry for the pause, I accidentally skipped this slide. Okay, so the ovaries then are the site of gamete production. That is the definition of a gonad. And it's actually suspended by these suspensory, ovarian, and broad ligaments. And so they're anchoring it to the pelvis, to the uterus, and to the floor of the pelvis. So the womb itself and the ovaries are all held into place by these ligaments. And so the follicles then that we mentioned before are the structural unit. They will have several different cell types, including that oocyte at the middle, but the fluid-filled cavity, the granulosa cells, which are helping to develop and serve a supportive role, the thecal cells, which is where the sex hormones are actually being synthesized, and the cumulus cells, so a type of granulosa cell that grows around and shields the oocyte from damage. Okay, moving on to oogenesis. It's going to be very similar to spermatogenesis in a lot of ways, but in this case, it's beginning in the fetal period. So in females, this stage is going on while females are still a fetus, whereas adult males are undergoing spermatogenesis. So the diploid stem cell is called an oogonia, and it'll divide by mitosis to produce those primary oocytes. And so just like before with the sperm, the primary oocytes have too many copies, so we need to undergo meiosis to produce the secondary oocytes, and then we need to undergo meiosis 2 to actually produce those ova. And so the end result then would be the 4 distinct haploid cells. Some differences between oogenesis and spermatogenesis. So in females, primary oocytes occur only in the fetus. So adult females already have all of the eggs essentially that they're going to have. And this meiosis process is actually arrested in late prophase 1 and resumes years later, if at all, due to puberty. And so in secondary oocytes, meiosis is arrested in metaphase 2. So basically all the chromosomes will be lined up at the metaphase plate, awaiting that equational division into those haploid cells. And so at birth, females are presumed to have their entire life supply of primary oocytes. Okay, scroll through here. So again, this is all happening while you are a fetus in your mother's stomach. So it's beginning during embryonic growth. All oocytes will be produced before birth, and as you progress and as you're ovulating these mature follicles out, the number of the primordial follicles will drop too. And so this development will pause until puberty. Remember, we talked about arresting during meiosis, for instance. And so the oogonium is the constantly dividing diploid stem cell, rapidly divide, producing millions of these undifferentiated stem cells that will eventually develop into the primordial follicles. And so around month 5 of fetal development, you'll have your max primordial follicle level, and they will begin deteriorating even before birth, but that will continue throughout the lifespan, significantly until the onset of puberty. So what is the relationship between the oocyte and the follicle? We saw in an earlier figure where the oocyte is actually located within the follicle itself, and basically it's all of the surrounding supporting cell types that we see. So the primary follicles have a single layer of the cuboidal pregranulosal cells surrounding the primary oocyte. As you move into the secondary follicles, so we've undergone some development here, there are now multiple layers of the mature granulosa cells. And in the vesicular antral follicles, also called a tertiary follicle, now there is a space, there's an open lumen, sort of fluid-filled cavity called an antrum. And so before this ovulation event, the primary oocyte, so still has, the undifferentiated chromosomes, inside the vesicular follicle, will resume that arrested meiosis and actually mature into the secondary oocyte. So here's the primordial follicle. Remember that maxes out around 5 months within the fetus, develops into the primary follicle, which now houses the mature oocyte. You have a zona pellucida, which is just a gelatinous covering. Now in the secondary follicle, we have this antrum, and as we move to the antral follicle, it will actually fill with a supportive fluid. So some histology: primordial, primary, secondary, early vesicular, and then finally we see the formation of the antrum. All right. So moving through the different stages of follicle development. Just in summary, you have the primordial follicle, which has the oocyte one layer of those pregranulosal cells, the primary follicle, your oocyte is going to enlarge, and actually the oocyte is the largest human cell type that we have. So several layers of those mature granulosal cells, and finally we see the zona pellucida starting to develop. In the secondary follicle, oocyte continues to grow, the cumulus cells develop, the cecal cells produce hormones, and moving on to the antral follicle, now we have the beginning of the antral development. Meiosis II will stop being arrested. It will proceed all the way into metaphase 2, so all the chromosomes, the sister chromatids, will be lined up at that plate. And then if fertilization has occurred, it will now proceed through meiosis II to form those haploid cells. And all that will be triggered, of course, hormonally. Post-ovulation, we call this the corpus luteum. And so the zona pellucida cells are actually going to secrete our estrogens, our progesterones and our estradiols. And if fertilization has occurred, it's either one or the other, then the corpus luteum will actually persist. And so you can actually still see it. If fertilization does not occur, then you'll see a corpus albicans. And so what happens is the zona pellucida will stop the secretion of those sex hormones, and your estrogens will actually drop off. And what's happening is the FSH and the LH are being secreted again. So it's one of the other. If fertilization occurs, then you'll see a corpus luteum. If fertilization does not occur, you'll see a corpus albicans. As mentioned before, this process is highly hormonally controlled. This process starts off, of course, with a couple of cycles that are actually feeding into each other. So the ovarian cycle is releasing an oocyte once per cycle, while the menstrual cycle is basically preparing the uterus to receive the oocyte. And so during each menstrual cycle, you have the endometrium, which is this highly vascularized layer, basically thickening and preparing for embryo implantation and development. Now, it's just like before where we saw a switch whether or not fertilization has occurred or not. So if it hadn't occurred, then the uterine lining is going to be sloughed off, and menstruation will occur, and you'll actually lose that inner layer. And then the cycle will just repeat until you get a fertilized egg. So ovarian cycle, roughly 28 days. Of course, this is just kind of an average of different people's cycles. But there are lots of events associated with the maturation of the egg. 2 consecutive phases, follicular and luteal, kind of named after the hormones controlling them, but period of vesicular follicle growth, so days 1 through 14 is the follicular phase. Then the luteal phase is days 14 through 28. Even though this is kind of a good average, only 10 to 15% actually of women actually have a 28-day cycle. So it does vary. So this follicular and luteal phase, the luteal phase will always be 14 days from ovulation to the end of the cycle. So what's actually going on then in these various phases? Well, if you look at the follicular phase, the antral follicles are becoming sensitive to that follicle stimulating hormone, as the name implies, and they are actually stimulated to grow. During the middle of this follicular phase, the FSH levels will drop, and one of the follicles will be chosen. It will outcompete the others and continue on in the process. The primary oocyte located within that follicle will then complete meiosis I, forming the secondary oocyte and first polar body. So what do I mean by that? Well, remember that in terms of chromosomes, females have 2 X chromosomes. Well, you really only need the genes from one, so you turn the other off in a mechanism called dosage compensation. And so what happens is one of those will basically turn into a polar body and condense down into a non-transcribable state. So moving into the luteal phase. So after ovulation, that follicle will collapse, and the antrum, that fluid-filled cavity, will actually clot with blood. The granulosal and thecal cells, remember the sort of protective granular layer, and the thecal cells, which are hormonal cells, those are going to enlarge and basically form a corpus luteum. This luteum will secrete the progesterone and the estrogen, so basically turning into synthesizing cells that will continue pushing out the sex hormones. So if we kind of come to another fork in the road here. If no pregnancy occurs, the corpus luteum will degenerate into the corpus albicans. If a pregnancy occurs, then the corpus luteum will continue to produce the hormones that will sustain the pregnancy until the placenta takes over. So, moving through this ovarian cycle, primary follicle, secondary follicle, vesicular follicle, ovulation, so now released into the endometrium here. This will fill with clotted blood and turn into the corpus luteum. Either if fertilization does not occur, then this will regress into a corpus albicans. If fertilization does occur, the corpus luteum will persist and continue to synthesize hormones. So let's talk about some of the hormones involved in this process. Our first is stimulated by the hypothalamus is the GnRH, or gonadotropical releasing hormone. That will stimulate FSH and LH secretion. FSH stimulates granulosa cells to release estrogen. The LH stimulates the thecal cells to produce androgens, and then these androgens can be very easily converted into estrogen. So things like testosterone and estrogen, very structurally related, and they can be kind of introverted. The follicles, as they continue to develop, will continue to synthesize the estradiol, and this will continue to rise throughout the follicular phase. After that follicular phase, the estradiol will actually fall off, begin to rise steeply. The FSH and LH blood levels will increase, and after the final maturation of the follicle, you get the release of the secondary oocyte. The luteophase will follow ovulation. So we've completed our follicle stage here. And via negative feedback loop, the secretion of LH and FSH will go back to their normal low levels. And this is caused by the GnRH back here. So negative feedback loop, we're going all the way back up to that hypothalamus. And the low gonadotropin levels will cause the corpus luteum to disintegrate. And as a result, you get a decline in estradiol and progesterone. So that's what that looks like. Basically, as we move through the various phases, you're going to have a push and pull of these various hormones, basically controlling this process. So let's talk a little bit about ovulation. Remember that ovulation is releasing that ovum towards the fallopian tubes, and it's being moved along by fluid movement. The ovum is being moved to the uterus, basically the end goal being the implantation of the ovum into the endometrium of the uterus itself. There's actually no direct connection between the ovaries and the fallopian tubes. They're actually just kind of sitting here loose underneath, so sort of inferior. So let's talk about that uterus then. So the uterus has 3 layers, basically an endometrium, which we had talked about before. It's highly vascularized. This is where the ovum is going to penetrate and actually receive its nutrient from. There is a myometrium, which is a very thick layer of smooth muscle for very strong contractions during birth. And then there's a outer covering, a epithelial covering called the parametrium, which is going to help lubricate the outer surface of the uterus and sort of reduce any friction on the outside. So then we have the cervix. This is a tapered entry and exit point, so basically connecting to the vagina, and then the vagina, of course, is serving as the birth canal, eventually leading out through this orifice. So the vaginal wall, of course, has sort of this texture to it for allowing for surface area and expansion, and it's very elastic to accommodate the relatively large skull sizes that human babies have. So females actually have separate urinary and reproductive outlets. Keep in mind that males do not. Of course, we share that sort of same urethra. And the external genitalia that you see are primarily protective structures. Okay, so that was the ovarian cycle. Now we're moving into the menstrual cycle. What happens to the endometrium and the ovum, essentially, depending on fertilization or not fertilization? Well, In the proliferative phase, we need to provide nutrients for the embryo. So that endometrium, that inner layer, is going to thicken up. This will be due to hormonal signals from the follicles itself, in this case estradiol, coordinated with the follicular phase. And you're going to see a secretion of nutrients during the secretory phase to help support the embryo. During the menstrual flow phase, that inner endometrial layer will be shed off, and this is in correspondence with growth of new ovarian follicles. And after about 500 of these cycles, human females are going to undergo menopause, and at this point, they will cease to ovulate and cease to menstruate. And so this thickness of this endometrial level is actually hormonally controlled and controlled by these cycles of the follicle development. So again, this is very much a hormonally controlled phenomenon. So you seem to see these sort of endocrine axes being in control here, where negative feedback is controlling some of these levels, whereas a very rare positive feedback mechanism is also controlling the production of certain of these hormones. We see that more, of course, after the embryo develops, but moving on. So, sex determination. What is it that makes a fetus male or female biologically? Well, it's actually determined very early on into development. Embryos are identical until that point, and as mentioned before, there is a portion of this development where essentially we are all sort of non-determined yet. The SRI protein found on the Y chromosome is actually what triggers male gonadal production. And there are some unfortunate cases where someone is born XY and they are insensitive to these androgen proteins, and so these individuals will present as biologically female, but they will actually have XY. So moving into development. So the developing offspring, of course, we call that concept. Pregnancy is the act of carrying a developed fetus, and then a gestation period for a human, usually around 280 days, we say 9 months. An embryo is conceptus from fertilization through week 8, and then a fetus is conceptus from week 9 through birth. And so we see some pretty big milestones coming about. The development of organs and the splitting off of germ tissues, going through these sort of primordial phases where we have like gill slits and stuff like that, tails, and then moving into the embryo, and then finally through the fetus. So fertilization, in simplest terms, is the combination of the chromosomes from the sperm with those of the oocyte to form a fertilized egg, also called a zygote. Before that can occur, the sperm must actually reach it. And so an oocyte is viable for 12 to 24 hours, and a sperm is viable to 24 to 48 hours after ejaculation. And the sperm must actually be capacitated before they can penetrate the oocyte. We used to think it was the fastest sperm that actually got there. That was the one that was selected, but actually there is some selection going on here with various receptors and stuff like that. But motility of the sperm will be enhanced. The cell membrane will become fragile, and hydrolytic enzymes will release that start to allow for the fusion of the two cells. Secretions within the female tract help it to weaken and thin out that acrosomal membrane. Remember, that's sort of the head of the sperm is covered in the acrosome, and we need to get rid of that. The sperm itself have primitive olfactory receptors that are allowing it to follow a chemical trail released by the egg and its supporting cell types. And so it's essentially sniffing its way to the oocyte. And so it needs to be capacitated by the chemicals within the female tract to actually get there. And so once the sperm has actually reached the oocyte, it needs to penetrate. So step one, weave through that corona radiata, that sort of outer layer. It will come in contact with that gel-like zona pellucida, and now the remains of the acrosome actually need to react. And so there will be a sperm binding receptor out in the zona pellucida. This will cause the release of calcium ions. The flow of calcium ions into the sperm will trigger the acrosome to react, and so digestion will begin on the zona pellucida. So a single sperm then will forcibly swim towards the oocyte membrane, and that oocyte has microvilli on the outside that will help to wrap around the sperm and kind of hold it in place. And finally, you get the fusion of membranes. The oocyte and sperm membranes fuse. The nucleus of the sperm, remember the nucleus has all the DNA, will enter the oocyte, and the tail and all the other sort of external portions are left behind. So here's that in a figure. The sperm binds to the zona pellucida. The contents of the acrosome are released by exocytosis. Hydrolytic enzymes help the sperm burrow through the zona pellucida, allowing for that binding and fusion. And then the nucleus will sort of insert itself into the egg cytoplasm. So what prevents multiple sperm cells from actually entering into one ovum? Of course it happens. Polyspermy occurs in some animals. Humans have monospermy. And we have a couple of mechanisms to ensure that happens. The first is a fast block. So once a sperm binds that receptor, the oocyte will actually turn off the other receptors. Then there is a slow block. Remember, some of this is due to calcium release entering sperm sites, and basically the plasma membrane around a successful sperm will start to release inhibiting proteins that basically destroy the sperm binding receptors and sort of down-regulate the ability of the sperm to fuse with the egg's outer covering. So going through these figures, if we look at the ovum structure here, you have that gel-like zona pellucida. It has receptors on the outside. Only capacitated sperm can bind, and the acrosome is going to begin to release these hydrolytic enzymes that help to dissolve that outer layer. The next sperm will attach to that dissolved region. More of the zona pellucida is dissolved, and then dozens of sperm are actually necessary to burrow through this one area. And basically it has to be actively inhibited every step to ensure that only the strongest and most successful get through. So there's an element of natural selection to which sperm actually makes it through. And then once a sperm successfully makes it through, you need to block the rest. So the fast and the slow blocks include turning off and shedding those receptors versus deactivating the receptors with some type of enzyme. So moving on to meiosis II and fertilization. First, you have a calcium surge, basically triggering it's time to undergo meiosis II. And this is what actually turns on the ova and turns the second X into a polar body. Remember, at this point, meiosis II has been arrested in an earlier stage. The ovum nucleus will swell to become a pronucleus, in this case, the female pronucleus. The sperm nucleus will swell to the male pronucleus, and then the DNA in each of these is going to replicate. And basically, as they get closer, the two pronuclei, a mitotic spindle made out of tubule proteins will form between them. Their nuclear envelopes will dissolve, and the chromosomes will be released in the mitotic spindle, essentially. They'll be pulled together by that mitotic spindle, combining them, forming the diploid zygote. And so the moment this happens, we call that fertilization. So here are the two pronuclei. The female pronuclei is noticeably larger than the male pronuclei. Here are those deactivated X chromosome containing polar bodies. And then we're going to move through the developmental states from zygote to blastocyst. So the embryo will travel through the uterine tube to the uterus. It's actually going to float freely there until it gets implanted. And you're going to get cleavage of the cell as well as blastocyst formation. Rapid mitotic division. So now our zygote is really trying to put off a lot of copies of those different chromosomes. So after 72 hours, the morula is going to develop with 16 or more cells. So we're moving quite fast now. Around 4 to 5 days, you'll have 100 cells in the blastocyst. It's starting to fill with a fluid in this sphere, and you're going to see differentiation into trophoblast cells. And these trophoblast cells will actually trigger development of placenta formation. Remember, we're sort of subsidizing the hormonal triggers here until that placenta can form and take over that job. So the blastocyst is going to float there for about 2 or 3 days, and then it's going to implant. Implantation has 3 steps: apposition, adhesion, and invasion. So apposition is when the blastocyst comes into contact with the site of implantation, basically the endometrium. During adhesion, the trophoblast cells will attach to the epithelial layer of that endometrium, and then during invasion, they will cross the endometrial epithelial basement membrane and basically invade that endometrial stroma. You might think, well, why would it want to do that? But remember that this endometrial layer is highly vascularized and thus can deliver a lot of blood flow and a lot of nutrients to what is essentially you're going to turn into your baby eventually. So the blastocyst reaches the uterine wall, the trophoblasts begin digesting the lining, and then that inner cell mass is going to differentiate with the endoblast cells forming first. So that cavity will form the amniotic cavity, and then you start to see the developing yolk sac, which will help to nourish the growing embryo in the future. So hormone levels are changing during all of this process. For one thing, you need to be able to tell your body not to continue to ovulate once fertilization has occurred. Otherwise, you could continue to get pregnant. So one thing that allows us to block that from happening is this human chorionic gonadotropin, HCG. It's basically going to tell the corpus luteum that since fertilization has occurred, you need to continue to secrete progesterone and estrogen, because these sex hormones will actually prevent future menstruation. Right? We've successfully implanted the embryo. We don't want to slough it off in our menstruation. So and basically this also stops from implantation of future follicles. So extraembryonic membranes, looking at the various membranes involved here around the embryo as it grows, you have the chorion. The chorion is highly vascular. It allows for a lot of exchange of nuclear and nutrient and waste products. It's comprised of the trophoblast and the mesoderm. So now we're starting to move into these differentiated germ layers. Early embryos have a yolk sac, just like a bird's egg does. And they also have an amniotic sac. The amnion is actually a protective layer. It's fluid-filled, basically just helps insulate the embryo from friction against the inside lining and basically blunt force trauma. Then you have the allantosis, which forms into the umbilical cord eventually, but is essentially separating the waste out from the embryo. As the placenta develops, it will be derived from both the chorion, this vascular sac, and the allantosis, which eventually splits off into the umbilical cord. So that's what that looks like. Here's the chorion, the vascular sac. The amnion is that outery water layer that helps to insulate. The allantosis stores waste until it can be removed via the umbilical cord. And the yolk sac has the initial energy source for energy. So here is a 3-month-old fetus. Here's a 5-month-old fetus. And then showing us a pathway of development here. You can see early on in the embryonic stage, we still have a tail. We probably still have the gill slits of our fishy ancestors, our reptile ancestors. But as we progress, the organs will begin to form, the genitalia will begin to become recognizable. In week 16, of course, the brain is going to begin advanced growth, the nervous tissue of our sensory organs. The GI tract is going to begin to secrete stuff. During week 20, growth is going to really take off, and so the fetus is going to move into the fetal position. voluntary muscular control will develop. Moving on, we'll have weight increases, blood cells, now from fetal production instead of the mother. And so around 28 weeks, if you have NICU intervention, you can actually make a viable fetus. And so around weeks 30 and 40, now the lack of room within the uterus is beginning to stress the fetus, and the lung surfactant of the type 2 alveolar cells is produced, basically allowing the lungs to function well and not stick to each other, and then the skin will finish developing as well. So our fetus has developed, now we need to get it out. So initiation of labor starts with the stimulated release of oxytocin from the posterior pituitary. Now if you remember back from the endocrine system, the posterior pituitary does not actually synthesize anything, it just stores what the hypothalamus makes. So oxytocin made in the hypothalamus, stored in the posterior, and during the initiation of labor is released. What this causes is gap junctions in the uterine muscle. So gap junctions are junctions that allow for the passage of material, including ions, which can signal. And so it's allowing for uterine muscle to start to make rhythmic contractions. These are the Braxton Hicks contractions. They will also soften that outer layer of the cervix by releasing prostaglandins. So prostaglandins are like a steroid-based, lipid-based inflammatory molecule, really, but it's softening the cervix and ripening it up in order to begin the process of moving this big fetus with a big cranium through. So oxytocin release is one of those rare positive feedback mechanisms that we have. But as the cervix begins to distend, what will happen is more oxytocin will be released, more prostaglandin will be released. So the cervix itself will continue to sort of thin out, and the oxytocin will allow for more rhythmic contractions to occur, leading to greater contractile force. This stresses the fetus out, and so more cortisol, a stress hormone, is being released, and that itself is a trigger. So greater distension of cervix, greater release of oxytocin, and this positive feedback mechanism is going to continue until the baby is born. So when you look at the relative size of the uterus before conception and during pregnancy, obviously the uterus itself has to stretch essentially, and it will stretch from below the navel to all the way up of the xiphoid process of the sternum. So that is the end of our reproductive system. Have a good day.